Hello everyone, welcome to 10-Minute Physiology. In today's video, we're going to be talking about the renin-angiotensin-aldosterone system. Specifically, we're going to be focusing on renin today. So with that, let's give it a go. So the first thing that I would like to do is to give you an overview of what stimulates the release of renin. So the main activator of renin release is going to be low blood pressure. So low blood pressure is going to primarily work through three mechanisms in order to increase renin release. The first mechanism is going to be the activation of the afferent arterial baroreceptors. So the activation of the afferent arterial baroreceptors is going to increase the release of renin from the juxtaglomerular cells. So the second mechanism is that when we have low blood pressure, this is going to decrease the amount of sodium chloride that arrives at the macula densa. When we decrease the amount of sodium chloride that arrives at the macula densa, this is going to increase the release of renin from the juxtaglomerular cells. And then the third mechanism is going to be the activation of the high-pressure baroreceptors. So the activation of the high-pressure baroreceptors is basically going to activate the renal sympathetic nerves, and when the renal sympathetic nerves are activated, this is going to release norepinephrine onto the juxtaglomerular cells, which will increase the release of renin. So these three mechanisms are the main ways in which low blood pressure is going to activate the release of renin from the juxtaglomerular cells. So now that we have this basic understanding, let's take a closer look at the cellular mechanisms behind how renin release is going to be regulated. So let's start off by drawing our juxtaglomerular cell. So the first thing that we're going to look at is we're going to look at the effect of high blood pressure on renin release. So high blood pressure is going to open or activate the calcium channels present on the juxtaglomerular cells. So when the calcium channels open, this allows calcium to flow into the cell. Now, calcium has two main effects in the juxtaglomerular cell. The first effect is it's going to inhibit the adenylate cyclase enzyme. Now, adenylate cyclase is an enzyme that converts ATP into cyclic AMP. So when calcium comes into the cell, calcium is going to inhibit this enzyme and therefore decrease the amount of cyclic AMP production. In addition to this, calcium is also going to stimulate another enzyme, specifically phosphodiesterase 1. So phosphodiesterase 1, in order to understand its effect, we have to know what cyclic AMP does in the cell. So cyclic AMP is going to activate an enzyme called protein kinase A. So protein kinase A is incredibly important for vesicular fusion. So what it does is it basically activates the proteins that are responsible for vesicular fusion and therefore allows renin release to take place from the juxtaglomerular cells. So in other words, if you activate pKa, you activate renin release, or you increase the amount of renin release that takes place. So in order to activate pKa, you need cyclic AMP. So basically, the effect of phosphodiesterase 1 is it basically converts cyclic AMP into AMP. And as a result, phosphodiesterase 1 decreases the amount of cyclic AMP. So what we see here is that high blood pressure decreases renin release through two mechanisms. The first mechanism is that through the entry of calcium, calcium inhibits adenylate cyclase, which decreases cyclic AMP. And calcium also activates phosphodiesterase 1, which decreases cyclic AMP. Now, when we decrease cyclic AMP, this decreases the activation of protein kinase A. And when we decrease the activation of protein kinase A, this decreases the amount of vesicular fusion that takes place. And when we decrease the amount of vesicular fusion, we decrease the release of renin. So high blood pressure decreases renin release. So now let's take a look at another substance, and this substance is going to be released from the macula densa. So this substance that we're going to look at is nitric oxide. So nitric oxide can diffuse into the juxtaglomerular cell, and when it does so, it activates an enzyme called soluble guanylate cyclase. So soluble guanylate cyclase converts GTP into cyclic GMP. Now, cyclic GMP is going to activate another enzyme called phosphodiesterase 3. Phosphodiesterase 3 converts cyclic AMP into AMP. And as a result, it decreases the amount of cyclic AMP. And once again, when we decrease the amount of cyclic AMP, we decrease the activation of pKa. And when we decrease the activation of pKa, we decrease the amount of vesicular fusion and decrease renin release. So nitric oxide released from the macula densa decreases renin release. So let's look at another substance that is released from the macula densa. So this substance is going to be prostaglandin E2. So prostaglandin E2 is going to interact with a G protein coupled receptor that is coupled to a GS protein. This GS protein stimulates adenylate cyclase, which increases cyclic AMP, which increases pKa activity, which therefore increases vesicular fusion and increases renin release. So prostaglandin E2 release is going to increase the amount of renin release. So now let's look at another substance that is released from the macula densa. And this substance is going to be adenosine. So adenosine is also going to interact with a G protein coupled receptor. However, this G protein coupled receptor is coupled to a GI protein. The GI protein is going to inhibit adenylate cyclase, and this will decrease the amount of cyclic AMP, which will decrease the activation of pKa, which will decrease renin release. So adenosine is going to decrease the amount of renin release. Now, another thing that we're going to look at is going to be the renal sympathetic nerves. So when the renal sympathetic nerves are activated, this is going to release norepinephrine. Norepinephrine is going to bind to GS-coupled protein receptors, and these GS-coupled protein receptors will activate adenylate cyclase, which increases cyclic AMP, which increases the activation of pKa, which increases renin release. So the activation of sympathetic renal nerves, which release norepinephrine, is going to increase renin release. Now, the last substance that we're going to look at is going to be angiotensin II. So angiotensin II is going to interact with G protein coupled receptors on the juxtaglomerular cells. The angiotensin II is going to activate these G protein coupled receptors, which activates the GQ protein. So the GQ protein is going to stimulate phospholipase C. Phospholipase C will convert PIP2 into IP3. IP3 will then move to the endoplasmic reticulum and bind to IP3 receptors, which will allow calcium to flow into the cytosol. The calcium will then inhibit adenylate cyclase, which decreases cyclic AMP, which decreases pKa activation, which therefore decreases renin release. In addition, the calcium will also activate phosphodiesterase 1, which decreases cyclic AMP and decreases renin release. So in addition to IP3, phospholipase C will also produce DAG from PIP2. So DAG is going to activate protein kinase C, and protein kinase C is going to inhibit vesicular fusion and therefore decrease renin release. So angiotensin II is also going to decrease renin release, and this provides a negative feedback mechanism. So in summary, here is a table telling you the effects of all the different substances. So high blood pressure decreases renin release. Nitric oxide decreases renin release. Adenosine decreases renin release. Prostaglandin E2 increased renin release. Angiotensin II decreases renin release. And renal sympathetic nerves, which release norepinephrine, increase renin release. So now I want to talk about the effects of pro-renin. So pro-renin can actually have effects on the principal cells in the collecting ducts. So the pro-renin is going to interact with the pro-renin receptor. Now, we're not going to talk about all these individual parts, but what pro-renin does is when it binds to its receptor through a signal transduction pathway, it is going to increase the amount of cyclic AMP in the cell. This increased cyclic AMP will activate protein kinase A, and then protein kinase A activates CREB proteins. So CREB is a transcription factor which is going to increase the transcription of aquaporin 2. The increased transcription of aquaporin 2 will basically increase the translation of aquaporin 2, which will therefore allow more aquaporin 2 proteins to be made. The aquaporin 2 proteins are then implanted into the apical membrane, which allows more water reabsorption to take place in the collecting ducts. So of course, the most important function of renin is going to be the cleavage of angiotensinogen into angiotensin 1. So renin is incredibly important because angiotensin 1 can then be converted by the ACE enzyme into angiotensin 2. Angiotensin 2 is going to have effects on the nephron. Then angiotensin 2 can stimulate the release of aldosterone. So renin is incredibly important for the renin-angiotensin-aldosterone system. And we'll talk more about this system in future videos. So I hope this video helped you understand the regulation of renin release, some of its effects, etc. And I hope to see you in the next video. Thank you for watching, and I hope to see you next time.